everyone. When we last left off on Tuesday, we were finishing up looking at the notion of what the electron transport chain is. It's certainly the one that's really the big finale, if you will. It's where most of your ATP actually arises from. But the couple things worthy of note before we get into a deeper dive, it occurs on the inner membrane space. It involves the flow of electrons, and it generates most of your cell's ATP. Now, the inner membrane contains these proteins that carry electrons, and what they do is they take those electrons from NADH, from FADH2 that were produced in glycolysis, that were produced in the Krebs cycle, and they create NAD and FAD that can then be returned for the glycolysis and Krebs cycle to utilize. Now, as the electrons move, as they transport through, there's a small amount of energy released as they get passed from protein to protein to protein. Now, this energy that's released helps move hydrogen ions into the intermembrane space. Why is that important? Because we now need to do something to oxygen. Oxygen is the final electron acceptor and gets reduced to form water. We need oxygen. So these hydrogen ions combine with oxygen and electrons to produce water. Now, if there was no oxygen, if there was none, this would cause the system to back up. You've got to have some all the way back to glycolysis because NADH and FADH can't be recycled. You don't take an oxygen, you get no ATP. No ATP, cells die, you die. Oxygen is needed for this process. Now, also contained in this is the process of chemiosmosis. Chemiosmosis is what produces most of the ATP for cellular respiration. In fact, for every turn, we get 32 ATP produced for chemiosmosis. Now, what does it require? It has to have hydrogen ions. It has to have an enzyme, a channel, which again is found in the inner membrane. Now remember, the electron transport is actually causing hydrogen ions to build up in the intermembrane space. Now, they can't diffuse back into the matrix. In fact, the channel, the ATP synthase channel, is the only place that hydrogen ions will flow. Now, as the hydrogen flow back out into the matrix through this channel, energy gets released that in turn produces ATP to the tune of 32 molecules, ATP molecules, for every glucose molecule you start with. So again, you can see this illustrated in the electron transport chain, continuing to bring hydrogen in that is used to form water. This energy feeds an ATP synthase, and as it turns, it generates ATP. In fact, 32 of them, as we already mentioned. So again, we see this here through let's think about it in terms of summary points. Glycolysis in the cytoplasm, glucose breaks down to 2,3 carbon molecules, getting a little bit of ATP, but it doesn't need oxygen. Step 2, if you will, the Krebs cycle. In the matrix of the mitochondria, pyruvate oxidation makes acetyl coenzyme A, releasing carbon dioxide. Now, this is all inside the mitochondria. The Krebs cycle then kicks in, followed by the electron transport chain, all of which yielding valuable products, all of which yielding valuable ATP. So if you look at it, the NADH carries the electrons gained from food to the electron transport chain. These electrons are passed along, and it pumps hydrogen ions across the membrane, which are finally accepted by oxygen molecules, producing water. So we can see what's going on here with this. In terms of total ATP, ATP from glycolysis, 2 molecules. ATP from the Krebs cycle, another 2. Another 32 from chemiosmosis and electron transportate, yielding a total of 36 for each molecule of glucose that enters. So in essence, glucose enters, goes through glycolysis, yielding 2 ATP. It also converts NADH to FADH2, producing more ATP. Pyruvate, going to acetyl coenzyme A, another 6 ATP. Acetyl coenzyme A enters the Krebs cycle, producing another 2, 18, and 4 respectively, yielding a total of 36 at the end. So if you look at aerobic cellular respiration, there's 4 stages: glycolysis, pyruvate oxidation, Krebs cycle, electron transport chain, chemiosmosis. And whereas pyruvate oxidation occurs in the mitochondria and carbon dioxide is cleaved off its waste molecules, the leftover molecules, the 2-carbon acetyl group, forms acetyl coenzyme A. Again, we see this here, the reaction proceeds, entering the Krebs cycle, they elect all these things there, these summary things yield energy that are used for cellular processes. But sometimes in a low oxygen environment, respiration must proceed anaerobically. In fact, our bodies sometimes do that when we're running oxygen deficit. So let's take a look, folks.
You are running a 1500-meter race. Breathing deeply to get the oxygen so that you can respire and keep up the energy supply to your muscles. Your muscles keep wanting more and more energy, but your lungs can't keep up. Do you have to quit the race? No. In the respiration video, we discovered the difference between aerobic and anaerobic respiration, and then looked at aerobic respiration in more detail. Sometimes animals and plants can't get enough oxygen to respire aerobically, such as during intense exercise, but they still need to respire to survive. After all, everything relies on respiration for energy. Luckily, there is a backup plan: anaerobic respiration. In this video we are going to have a look at anaerobic respiration in both animals and plants. Starting with animals. A cheetah sprinting to catch some prey cannot take in enough air and blood around the body fast enough for aerobic respiration. The heart and lungs can't keep up, so anaerobic respiration is carried out instead. Here is the generalized equation for anaerobic respiration. See how it differs from aerobic respiration. There is no oxygen involved in anaerobic respiration. It is much less efficient than aerobic respiration, and much less energy is released. This is because the glucose is only partially broken down. Another problem is that lactic acid is produced. This is actually a poisonous chemical that if it builds up in the body, the muscle stops working and you get a muscle cramp. You can only get rid of lactic acid by taking in oxygen again, and thus replacing the oxygen there. Oxygen is needed to break down lactic acid, turning it into carbon oxide and water. You know how you breathe deeply after intense exercise? This is your body replacing the built-up oxygen debt. So that is how anaerobic respiration works in animals. What about in plants? The oxygen supply can run out for plants too, such as in waterlogged soils. This then forces plants to have to carry out anaerobic respiration, as they too need to respire constantly. Here's a generalized anaerobic respiration equation for plants. Notice how instead of lactic acid being produced, ethanol and carbon dioxide is made instead. In yeast, this process is called fermentation, and it is used to bake bread and brew alcohol. Ethanol is the alcohol found in beer and wine, for example. And in bread making, it is the carbon dioxide that is useful. The bubbles in carbon dioxide help the bread rise by expanding the dough. So you should now know that when our heart and lungs cannot keep up with the oxygen demand, or plants do not have access to sufficient oxygen, we have a B plan, anaerobic respiration. This is when the glucose is broken down directly, but it produces much less energy than aerobic respiration, which is why it's the second choice. And in animals it produces lactic acid as a byproduct, which causes muscle cramps, and so we end up with an oxygen debt which needs repaying after exercise has stopped.
So if you're looking at anaerobic respiration, anaerobic means without oxygen. Now, no oxygen, NADH and FADH2 can't get rid of their electrons, remember. As a result, no NAD for glycolysis or the Krebs cycle. So what happens? Well, NADH and FADH2, they got to get their electrons to something else instead of oxygen. And if they're able to do so, this allows NAD and FAD available for glycolysis, which can allow energy production to continue. In yeast cells, the only one specific, probably the one we're most familiar with, anaerobic respiration produces ethanol, fermentation. It's how beer gets made. It's how various alcoholic beverages, bread making even. So what the process involves, again, we start with glucose still. We got to have that sugar to start with. Glycolysis proceeds, yielding NADH and ATP. The difference, though, is that in the second phase, pyruvic acid enters, carbon dioxide gets released, which is what causes bread to rise, the bubbling, NAD is produced, but instead of acetyl coenzyme A, we now get a 2-carbon ethanol molecule. Or, let's take a look once again.
When I was a kid, swimming was one of my favorite things to do. I was on the swim team starting when I was 4 years old and then on and off throughout grade school. Although I wasn't especially fast. I just loved the water. I used to dream that there was a way I could be like a fish. When I was little, I had a misconception that fish didn't need oxygen. Later on, I learned that no, most fish have gills that allow them to extract the oxygen, which they need, from the water. So then I just thought it'd be really cool if I had gills. But alas, no gills for Pinky. Oxygen is a really big deal. So many organisms, from fish to plants to humans, need oxygen. And yes, even though plants make oxygen in photosynthesis, they still perform cellular respiration, and therefore plants still need oxygen themselves. There's often a misconception that plants don't need oxygen. That's just not true. So why do these organisms need oxygen? It's similar to why you need oxygen. If you've ever wondered why you need to breathe, which is done by the respiratory system in your body, zoom into the cell level. Cells in your body use the oxygen you inhale to perform cellular respiration. The formula here requires inputs, otherwise known as reactants, to make ATP. And oxygen is one of those reactants in the overall equation that is needed to break glucose down in forming ATP. Why ATP? ATP stands for adenosine triphosphate, and it's action-packed with 3 phosphates. It has the ability to power many cellular processes. Typically, it's coupled to other things that it may be powering. Now, after losing the phosphate, the molecule is ADP, adenosine diphosphate, because it has 2 phosphates. In cellular respiration, though, there are enzymes that can add another phosphate to it and convert it back into ATP again. This particular formula of cellular respiration is aerobic, meaning overall it requires oxygen. It's pretty complex, and we have a video breaking down the steps. But that's not what this video is about. This video is about what happens when there is no oxygen, because cells still need to make their ATP. So what kind of cells can handle the no oxygen thing? Well, many types of bacteria can, many types of archaea, yeast, which is a fungus that could be helpful, like making your bread rise, your muscle cells can, for a while anyway, and these are just a few examples. Now, these organisms handle the lack of oxygen in different ways. Some organisms, such as some types of bacteria or archaea, can do anaerobic respiration. They can continue to perform glycolysis, Krebs, and the electron transport chain, just like aerobic cellular respiration. But since there is no oxygen to be that final electron acceptor at the end of the electron transport chain, they use something else. Sulfate, for example. These organisms are specifically adapted to be able to use a different electron acceptor in this anaerobic respiration. Another option is the organism may just stick with doing glycolysis, which doesn't require oxygen, and then the addition of some way to get their NAD plus back. We'll talk about what that means in a minute. This process is called fermentation, and that's what we're going to focus on. Fermentation is a way to be able to handle the little to no oxygen issue. It allows for glycolysis to happen and for glycolysis to keep going. That means making ATP where there is no oxygen. And while you won't make as much ATP in this process as you would aerobic cellular respiration, you can't be too picky when oxygen isn't around. Okay, so recall what glycolysis is from our cellular respiration video. In glycolysis, you take glucose, a sugar, and it gets converted into pyruvate. This takes a little ATP cost to actually start it up, but overall, per glucose, you make 2 net ATP molecules, and you also produce 2 NADH. Recall that NADH is a coenzyme and an electron carrier. We also need to mention that NADH didn't just poof appear as a product. No, because NAD plus actually was reduced to NADH when it gained electrons. And if the words reduced and oxidized are confusing, you can remember the famous Leo Gurr mnemonic. Lose electrons oxidized, gained electrons reduced. So NADH to NAD plus is oxidation because it loses electrons, and NAD plus to NADH is reduction because it gains electrons. Now, NADH, the electron carrier, would normally be delivering the electrons gained to the electron transport chain, if this was aerobic cellular respiration. Once losing their electrons, NADH would be oxidized into NAD plus and be ready to be involved all over again in glycolysis. But there's no electron transport chain in this fermentation process, so we've got to regenerate the NAD plus somehow. NAD plus is needed here, after all, for glycolysis to continue. Fermentation, therefore, adds another little step to the end of glycolysis, a step to help regenerate NAD plus. This happens because fermentation allows NADH to give its electrons to an electron acceptor, which in the 2 fermentation examples we are going to give will either be a derivative of pyruvate or pyruvate itself. So here we go with 2 types of fermentation, which both result in different products from pyruvate. Alcoholic fermentation is done by some types of yeast. So first glycolysis happens, which yields 2 net ATP, 2 pyruvate, and 2 NADH. Now, we need this step to regenerate the NAD+, so we can keep doing glycolysis. The 2-pyruvate is used, which will ultimately produce carbon dioxide and 2-ethanol, alcohol. But the derivative of pyruvate shown here, acetaldehyde, can act as an electron acceptor in this process, so the 2-NADH can be oxidized to 2-NAD+, so that glycolysis can start all over. Since alcohol is a waste product in this process, this is why many yeast are used to produce alcoholic products. Yeast also can do alcoholic fermentation in making bread, and the carbon dioxide product we mentioned is involved with helping the bread rise. The tiny amount of alcohol produced in the short fermenting time of bread will evaporate in the baking process. Lactic acid fermentation, as done by cells such as your muscle cells, for example. While your muscle cells can do aerobic cellular respiration, they can shift to lactic acid fermentation if they experience an oxygen debt. This could happen if you're working out very intensely, where your blood is unable to deliver a sufficient amount of oxygen to them for their demand. Just like alcoholic fermentation, we start with glycolysis, that yields the 2 net ATP, 2 pyruvate, and 2 NADH. But now we need this step to regenerate the NAD+, and this step is a bit different from alcoholic fermentation. The 2-pyruvate on the reactant side will ultimately yield 2-lactate. The pyruvate can act as an electron acceptor, allowing NADH to be oxidized to NAD+, so that glycolysis can start all over. By the way, this lactate product, or specifically its other form, lactic acid, has often been blamed for the muscle soreness that occurs after intense exercise. In many of my teaching years, this was often discussed, but there's some recent research that may contradict that. Science is so cool. Check out our further reading suggestions and our video details to learn more. Lactic acid fermentation is also done by bacteria that are involved in making yogurt, and lactic acid can contribute to its sour taste. So overall, fermentation is a pretty remarkable process. Although, it does make us appreciate oxygen because despite how absolutely awesome fermentation may be, it just can't make as much ATP as aerobic cellular respiration. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
So that's an overview of anaerobic respiration. Of course, there's couple different pathways. Of course, we have alcohol fermentation, commonly utilized by plants, yeast, and so on. Is that here, the NADHs produced during glycolysis pass their hydrogen onto acetaldehyde. How is acetaldehyde formed? Is when carbon dioxide is removed from pyruvate by pyruvate decarboxylase, as we see in the diagram below. So again, glucose goes in, ATP comes out, producing pyruvate. That's glycolysis. Note that no action required. No change. Occurs the same regardless. In the absence of oxygen, however, well, it's not an option. You got to do something. Keep things going. So water enters, carbon dioxide comes out. This yields a 2-acetaldehyde, again, modified. The electrons from NADH, hydrogen from the NADH combine to yield ethanol as a product. So ethanol is produced. Again, we have acetaldehyde form and acts as the proton acceptor, the electron acceptor. So in animal cells, though, as opposed to plants, we don't produce ethanol, we produce lactic acid. Now, and the cells aren't producing that. That's why you get what we call a stitch. Muscles become cramped, build up lactic acid. So when oxygen becomes available, lactic acid will then convert it back to pyruvate, which is why if you run, you get a cramp. You may stop running, you start breathing deep, and eventually the stitch goes away as the lactic acid gets broken down. This pyruvate, once produced, then continues on the Krebs cycle. Now, what it does do is it's useful because it provides a short burst of energy when oxygen is not available. But it can only produce a small amount of ATP compared to what you get aerobically. Much less efficient, but the byproduct is lactic acid, which is what causes your muscles start cramping up. So with lactic acid fermentation, normally, again, as I've already mentioned, we would normally get energy and glucose through aerobic means. But if we're exercising strenuously, our requirement outstrips our supply. We need more ATP than can be supplied by aerobic. Under such conditions, additional ATP gets provided by lactic acid fermentation, as you see here. So again, process is glucose molecules, a little bit of ADP, and ATP goes in, ATP is produced, pyruvate comes out, the electrons are accepted by the pyruvate, yielding lactic acid. Also part of the process, we get NADH oxidized to NAD, which allows for the continuation of glycolysis. So let's take a look.
Glycolysis is when we take glucose molecules and we do a bunch of chemical reactions, eventually forming pyruvate molecules. And for simplicity, let's say glycolysis is when we take glucose, we do a bunch of chemical reactions, eventually forming pyruvate molecules. And every single cell in your body right now is going through glycolysis. So why does every single cell in your body go through glycolysis? Well, when we go through glycolysis and take glucose molecules and convert them into pyruvate molecules, we form a net of 2 ATPs. So this is one of the ways our bodies produce these high energy ATP molecules, which we know are important for fueling all the energetic processes we need for life. So glycolysis produces ATP molecules. However, something important to realize is each of these steps requires its own enzyme. And this particular step requires an enzyme that requires an NAD plus to be converted into an NADH. So for this step to work and therefore allowing the cell to go through glycolysis, we must convert an NAD plus into an NADH. This is absolutely mandatory. So therefore, if we're doing glycolysis, if a cell is doing glycolysis to produce ATP, it's constantly converting these NAD pluses into NADHs. Every single time we do this step, we convert an NAD plus into an NADH. So if we keep on doing glycolysis to produce ATP, we're going to keep on converting these NAD pluses into NADHs. And the more glycolysis we do, the more NAD pluses are going to be converted into NADHs. And eventually, we're going to do so much glycolysis that we convert all of our NAD pluses into NADHs. So what happens once we convert all of our NAD pluses, all the NAD pluses into NADHs? Well, now we're going to have no more NAD pluses. So what happens once we have no more NAD pluses because they've all been converted to NADH? Well, now that we have no more NAD pluses, now this enzyme can't work. And if this enzyme can't work, we can't do glycolysis, so now we can't produce ATPs. So this is bad. We need to find a way to reform those NAD pluses. So normally, how do we reform those NAD pluses so we can keep on doing glycolysis to produce ATP? Well, normally what happens is we know the mitochondria has this electron transport chain. And this electron transport chain accepts electrons from NADH, converting it into NAD+. So this is normally the way we reform NAD+. Normally, the mitochondria is going through the electron transport chain and oxidative phosphorylation, and when it does is it converts NADHs into NAD+. So now we have more NAD+, so now we can keep on doing glycolysis to produce ATP. But in order for this electron transport chain to work, we need oxygen. We need a lot of oxygen to be the final electron acceptor of this electron transport chain. Because we know this NADH is going to donate electrons to the electron transport chain, which must donate those electrons to oxygen. So what happens if maybe we're in rigorous exercise, or maybe just for whatever reason we don't have enough oxygen? Well, if we don't have enough oxygen, we can't do the electron transport chain, and if the electron transport chain can't work, it can't accept electrons from NADH, so therefore we can't take that NADH and convert it into NAD+. So this is bad. So if there's not enough oxygen, how are we going to reform NAD+? Because we know if we want to keep on doing glycolysis to produce ATP, we need to reform NAD+, so we can do this step, so we can do glycolysis. So how are we going to create NAD+? Well, we're going to do fermentation. So fermentation, the way human cells do fermentation is we take pyruvate and we convert it into lactate. So why? Why go through fermentation, taking these pyruvates and converting it into lactate? Well, to do fermentation, we need this enzyme to function. And for this enzyme to work to allow this step to occur, it must take these NADHs and convert it into NAD+. So therefore, as we do fermentation and as we convert these pyruvates into lactate, we're taking NADHs and converting them into NAD+. So now we keep on doing fermentation and we keep producing these NAD+. And now eventually we're going to form enough NAD+, so now we've reformed NAD+. And now that we've reformed NAD+, now we can allow this step to occur. Now this enzyme can function, so now we can allow this step to occur. So now we can do glycolysis and now we can produce our ATP. And something important to realize is this is the primary reason why we do fermentation. This is the only reason why we do fermentation. So we can reform those NAD pluses. So now that we reform those NAD pluses, now we can keep on doing glycolysis to produce ATP. So this is specifically how human cells do this. And notice it's kind of like a cycle. We do this step to do glycolysis and we form these NADHs, but now we do fermentation to reform those NAD pluses. So now we can keep on doing glycolysis to produce ATP. And specifically, this is the way humans do fermentation. This is how humans specifically do this step, forming lactate, so it can reform these NAD pluses so it can do glycolysis. However, bacteria and yeast are a little different. So it's the same idea. They do glycolysis to produce ATP, but all their NAD pluses are converted to NADHs. So they need to find a way to reform NAD plus. So again, they do fermentation, but they do it a little differently. Essentially, they take their pyruvate and form acetaldehyde. Then once they form this acetaldehyde, they convert it to ethanol. So why do they do these processes? Well, again, this particular step of taking acetaldehyde and converting it to ethanol, this particular step does the same idea. It takes an NADH and converts it into NAD+. So now as these yeast or bacteria go through fermentation, they're forming NAD+, which can now allow them to keep on doing glycolysis to produce ATP. And just a little fun fact, other bacteria can go a step further, forming acetic acid. And notice, this is one of the way we produce ethanol and acetic acid. And notice, ethanol is drinking alcohol. This is drinking alcohol. Ethanol is what gets you drunk. And this is one of the ways we use yeast or bacteria to create ethanol. And acetic acid is vinegar. This is what's used to make vinegar. So this is, through fermentation, is how these products are made. And again, other types of bacteria can do other types of fermentation to produce other products. But the point is, if any cell is doing high amounts of glycolysis, they're using up all their NAD pluses. So therefore, they have to do some type of fermentation to reform those NAD pluses. So again, now let's talk about humans. So in humans and skeletal muscle, normally what's happening is we know skeletal muscle requires a lot of ATP to create those forces, to create forces for skeletal muscle to work and for movement. It requires a lot of ATP. So normally the way they make their ATP is they do a lot of glycolysis, and then they take that pyruvate to enter the mitochondria, which we know fuels the electron transport chain through aerobic respiration to create ATP. And in fact, this is through this aerobic respiration is where these skeletal muscles create the vast amount of their ATP. However, if this cell and if the body is going through excess exercise, maybe there's not enough oxygen. Maybe there isn't enough oxygen to get to the muscle cells, or maybe we're just doing so much aerobic respiration that we're using up all the oxygen. So during strenuous exercise, there's not going to be enough oxygen. So if there's not enough oxygen, we can't do aerobic respiration and fuel the electron transport chain. So therefore, we can't create ATP through this aerobic respiration. So during strenuous exercise, when there's not enough oxygen, how are we going to create all of our ATP? Well, now we have to do excess glycolysis. Now we have to do high amounts and high flux of glycolysis. So we have to do a lot of glycolysis to produce enough ATP to energize these cells. But we know if we do high amounts of glycolysis, we're using up all of our NAD pluses. All of our NAD pluses are going to be converted to NADHs. So therefore, we're going to have to do high amounts of fermentation. So skeletal muscle during strenuous exercise are doing high amounts of fermentation to reform those NAD pluses so they can keep on doing glycolysis to create their ATPs. But during the strenuous exercise, they're going to do a lot of fermentation, so they're going to create a lot of lactic acid. And this is an acid. This has a carboxylic functionality, which we know is an acidic functionality. So lactate is acidic, and lactate can enter the bloodstream, and it creates an acidic condition, and that's what causes muscle fatigue. So what are we going to do? If we do high amounts of glycolysis and fermentation, we're going to produce a lot of lactate. So what are we going to do with all this lactate? Well, this lactate is kind of a waste product. It's kind of like trash. So what are we going to do with all this trash? Well, we're going to send it to the liver, and we're going to tell the liver to take care of it. So what is the liver going to do with all that lactate? Well, they're going to take that lactate and enter it through gluconeogenesis. So when the liver takes the lactate and goes through gluconeogenesis, they form glucose. So what is the liver going to do with all that glucose? Well, now it's going to send that glucose to the skeletal muscle. So now the skeletal muscle gets that glucose. So now the skeletal muscle can use that glucose as a source of energy to keep on doing glycolysis to create the ATP it needs for the skeletal muscle to work, to apply all those forces and to allow the muscle to contract and work. So this process of doing high amounts of glycolysis and fermentation to form lactate, and then the lactate sent to the liver, and then the lactate enters gluconeogenesis to create glucose, and then that glucose is resent to the skeletal muscle. This cycle is referred to as the Cori cycle. And if you're interested, this was actually discovered by a woman. A woman discovered this, which is kind of a neat fact. But again, so this is referred to as the Cori cycle. And again, this is required for the skeletal muscle to have the energy to allow it to keep on applying those forces. So again, not only do skeletal muscles do a lot of fermentation, but also cancer cells. Cancer cells have a phenotype and where they do high amounts of glycolysis and fermentation, and this is referred to as the Warburg effect. But so cancerous cells are also doing high amounts of fermentation, producing a lot of lactate. So again, that lactate can be sent to the liver through gluconeogenesis. So now that glucose can be sent back to the cancer cells. So now the cancer cells can keep on essentially, now they have that source of energy so they can keep on creating ATP to allow the cancer cell to divide. So therefore, theoretically, if you were to block this Cori cycle, now the cancer wouldn't have glucose, so now it wouldn't have a source of energy. But it's a little more complex than that. But again, the point is, this is the Cori cycle. This, in this context with the skeletal muscle and the liver, is normally in the context you'll hear about the Cori cycle.
So again, there's been cases where, for various reasons, it's actually happened where mammals, in this case a human, was actually undergoing alcoholic fermentation, producing alcohol in his body, right? Sometimes bacterial infections can cause it because the bacteria are actually respiring anaerobically, producing alcohol. Now, in terms of exercise physiology, we see all these things. These are related. You need to take a point of reading through that section of your textbook, pages 225 through 227. So when you have heavy, rigorous exercise, because of the shortage, the body will switch over to anaerobic respiration. In fact, it's a good measure of a person's level of fitness. Where do they go? How much can they cope in low oxygen environments? Well, again, there's all kinds of reasons for that. A person might undergo VO2 testing. I'd suggest that you look at the videos that you see listed here, VO2 max. At NHL combines, they actually, as a measure of what the person's aerobic fitness is. How much oxygen can be removed? How quick are they respiring? How quick are they utilizing energy sources? Now, typically we get VO2 max values. They can get an idea. As we age, these things drop off. And there's also your lactic acid threshold. So the lactic acid threshold is when does your intensity as which blood lactic acid begins to increase sharply. Now, if you go beyond that, it might limit duration of exercise, pain, muscle stiffness, fatigue, athletic training. The idea is to actually improve circulation, how efficient you're using oxygen by decreasing the production but increasing the threshold. Ideally, well, an elite athlete, they've reached threshold at or above their 80% of the VO2 max. Untrained athletes, well, 60%. Big difference. Of course, in exercise physiology, of course, there's supplementation has been utilized. And again, this might have the benefit of producing more ATP, but it might also cause other chemical problems as we see listed. Sometimes we get metabolic poison, which actually interrupts the process. And again, just by way of an example, when you have cyanide, carbon monoxide, this interferes with the ability for the protons to be utilized. As a result, the electron transport chain gets interrupted, and this creates obviously problems in your ability to utilize and produce energy. And while this is a oversimplification, you know that coenzyme A enters in and actually NADH. These are all things that are part of the process which you already looked at previously. Alcoholic fermentation. See this illustrated on page 221. On page 224, we see the process of lactic acid fermentation, what it involves. Now, we need to utilize energy. Sometimes your body will start metabolizing fats, metabolizing sugars, as well as amino acids. Your body will use whatever carbon source it can to actually generate the energy that's needed. Of course, all of this allows for the formation of organic materials that's used to synthesize organic molecules that we need. So in summary, you see this listed here. It's a very important process. Thanks for paying attention.